Welcome, everybody. Um, so, what we're going to do today is I'm going to break down my story into three individual chapters. Now, many of you may be pretty well understood about the CAR T field, but I will begin with an overview and background of some of this for some of our folks that are joining us that are really trying to learn a little bit more about this technology. But I also want to talk a little bit more during this introductory period about cytokine release toxicity, because this is a very important aspect of this treatment strategy in developing new therapies to understand the balance between cytokine release and tumor efficacy responses. And then in chapter 2, we'll talk about some of the published examples of recent advances in chimeric antigen receptor therapy, and we'll include some examples for the CAR T space, CAR NK, and CAR M, and how these folks are designing these things to improve patient outcomes. And they're looking at things at dual targeting, affinity tuning, cytokine co-expression, and how some of these CAR Ms actually can modulate the tumor microenvironment, which is really interesting. And then in chapter 3, we're going to talk about a new platform that has been developed by the Jackson Laboratory that is really important because it can take a look at the association of tumor efficacy in terms of CAR T preclinical efficacy, but also activation-induced cytokine release and the systemic toxicity that can be associated with that, and allow drug developers to find a balance between efficacy and safety in a more reliable fashion. So, chapter 1, let's jump right in and talk about this new technology. So, CAR T cell therapy. T cells are genetically engineered ex vivo to express a receptor that allows them to target tumor cells. And there are at least 2 different FDA-approved CAR Ts, and these are targeted towards B-cell tumors. So they're applicable to NHL, CLL, ALL, diffuse large B-cell lymphoma, multiple myeloma, and folks are trying to get clearance for other forms of B-cell tumors all the time. The clinical success of these treatments have really stimulated folks to think about going beyond just B-cell and liquid tumors and thinking about myeloid tumors, solid tumors, et cetera. These chimeric antigen receptors are also being applied to other cell types beyond just T cells, as we mentioned a moment ago. But they not only include NK cells, but NK T cells and even gamma delta T cells, and of course the myeloid cell population that we've already mentioned. But what's different about liquid tumors and solid tumors is that these different CAR cells that are found in the bloodstream really need to leave the bloodstream if they're going to be able to address the solid tumor story. And then, of course, solid tumors have a tumor microenvironment that can be very immunosuppressive. And so that sets up a whole new different paradigm, and folks are taking interesting strategies to try to address that. And we'll talk a little bit about that in the slides coming up. Okay, so what's going on in these patients is that most of these patients have already received first and second line therapy in many cases. So the folks that are getting these CAR therapies typically have relapsed and refractory disease. So this is important for understanding the context in which these cells are being delivered. These patients have already experienced probably very high dose chemotherapy, sometimes in combinations with immunotherapies like rituximab. And then sometimes they'll bounce back from that, unfortunately, and require either autologous or allogeneic stem cell transplantation. So these patients have really been through a lot. And what's going on here is the patient T cells are collected by leukapheresis. They're genetically modified, expanded outside the body, and then reinfused. And this is what we're calling autologous CAR T cell therapy. And it's autologous because, again, we're using the patient's own T cells. And the reason that's important is because even though they're being genetically modified to express a certain receptor on the surface, in most cases, they're still expressing the native T cell TCR. So in other words, if you took T cells from another patient that's not HLA matched, when these T cells go in, they could have a response against the human tissues because they're be recognized as foreign, and they could develop graft versus host disease. So that's what we mean by autologous CAR T. Now, one of the things about autologous CAR T is this is an individualized treatment, and it is a very expensive procedure. So a lot of folks are taking a look at allogeneic or off-the-shelf CAR T. This is where you would deplete these cells of their native TCRs and introduce the CAR T molecules, but you also have to take additional steps to prevent the host immune system of seeing these allogeneic T cells as foreign. So that's kind of a complex, and it's kind of like the next level of CAR T therapies and CAR NK therapies. But for the purposes of this talk today, we're going to be talking really in the context of autologous CAR T cell therapies, CAR NK, CAR M, et cetera. So what exactly are these chimeric antigen receptors? Well, what they have is an extracellular binding domain with specificity to tumor-associated antigens, and it's typically made up of single-chain variable fragments, as shown here. So you can develop, using antibodies, a binding region, and then take that binding region and develop this variable fragment that can be covalently linked to a hinge structure, and that is an intracellular domain, typically CD3 zeta, which is a portion of the natural TCR, which is known to activate the T cell. So through binding, you get these T cell mediated synapse with the cancer cell, which allows structural changes that allows activation of the T cells, and the T cells respond in a cytotoxic manner against the tumor. Now, the first generation didn't have a costimulatory domain that was also engineered into this, but the second generations have CD28 or 41BB, and then the third generations that come along really include both of those costimulatory domains to enhance the activity of the T cells. And we'll come back to that a little bit more in just a moment. But there are a number of different factors impacting CAR function. One of those is tonic signaling, which is very complex in the sense that it is due to the perhaps overexpression. So if the expression of the CAR construct is too high, you can get receptor-mediated interactions that can lead to antigen-independent signaling and activation of the CAR T, or there can be interactions, as you'll see in a moment, between some of these hinge domains that can lead to this. There's also toxicity. Those toxicities include both tumor lysis syndrome, cytokine release syndrome. The cytokine release syndrome can lead to neurotoxicity. And if you do not design your binding region carefully enough, you can have what's called on-target but off-tumor toxicity. In other words, normal cells within the body that also express the tumor target, but which can be recognized by these CARs and lead to tissue and organ damage. In a pro-inflammatory environment, you can have antigen loss, so there's target limitations, or there could be very heterogeneous populations of the tumor, and all certain members of the cell population within the tumor are killed off. And then when we get to the idea of solid tumors, there's the tumor penetrance and trafficking to those tumors that can be important. If these CARs get overactivated, you can have activation and induce cell death, and the cells can kind of kill themselves off before they can even eradicate the entire tumor if there's too strong of a pro-inflammatory response. And then there's exhaustion. The tumor microenvironment is very important, particularly when in the context of solid tumors. You have upregulation of PD-1, TIM-3, LAG-3, these molecules that are normally present that are involved with tamping down the immune response, and people are using co-treatments to try to block those different pathways. So there's a lot of different things that need to be taken into account with these therapies. So we really need a good assay system for looking at all these different features. Now, when it comes to these co-stimulatory molecules that are part of these chimeric antigen receptors, they can really influence the T cell populations in terms of their longevity, durability, and their energy function. So 4-1BB, for example, really can lead to longer persistence and a greater frequency of central memory phenotypes. And these cells have a high mitochondrial respiration. If you use CD28 alone, you have tend to have a shorter persistence, greater frequency of T effector memory phenotypes, and that leads to them being shorter lived. And they have a higher glycolytic respiration. And generally speaking, when you include both of those co-stimulator domains, you have increased effector function coupled with superior expansion and persistence. Now, there's also other factors that are associated with the subcomponents of each of these CAR designs that can impact the function. Now, one of them is that this variable region here that's specific, depending on the binding affinity, can influence a lot of different things, and we'll come back to that with a very specific example later on. But these regions can also influence tonic signaling. These things can react with one another and have non-antigen specific activation of these T cells, decreasing the effectiveness of these molecules. And also these spacer molecules that are engineered into can influence what we call the immunological synapse. So the length of this influences the ability to bind to the tumor target within this membrane space, because remember, these things need to swivel around and move. There's other molecules in the synapse, and so that can have an influence on it. The transmembrane domain can be very important depending on the co-stimulatory molecules that you're using. And then we also talked about the intracellular domain and the inclusion of these co-stimulatory molecules and how you design that. So these are what are considered the CAR intrinsic factors, but there's also CAR extrinsic factors, which include the delivery system, whether you're using a lentivirus, an adeno-associated virus, electroporation, and there's things that are important about that in terms of where these things land in the genome. If they land in the genome randomly, they could insertionally inactivate genes. They could be downstream from strong promoters. They could be downstream from weak promoters, regions of the genome that suppress function. You could land in the middle of a tumor suppressor gene. So you're trying to get rid of cancer, but now you've gotten rid of a tumor suppressor gene. So the T cells that you're introducing itself can cause cancer. The promoter strength, which can lead to overexpression in tonic signaling. So all of these things need to be assayed in exquisite detail before you go into first-in-man studies. That's related to CAR T, but CAR NK also have very important vector design and structural designs. So in the top row here, we're showing the natural NK cell receptors. And what you'll notice here is CD16, which is expressed by NK cells, and associated with that is the CD3 zeta that we talked about just a moment ago that is also utilized in activating NK cells. So CD16 binds the Fc tail of monoclonal antibodies, and that's the pathway at which NK cells are activated through antigen or antibody-dependent cellular cytotoxicity pathways. So folks have designed these variable fragments to the tumor targets and engineered them with CD3 zeta. They've also engineered these to include components of these co-stimulatory domains of these natural receptors to get activity. And then in the second generation, they're combining these natural co-stimulatory domains that are associated with natural killer cells along with the CD3 zeta component of the arm. So you could see a lot of the similarities in technology between applying these receptors to these different cell types. But what we know from the clinical experience by using both through, these uh, safety trials as well as other clinical trials and then looking at the patient responses, we know that these cells get activated very strongly and they release cytokines and the patients have a strong CRS response. The incidence of CRS varies by the CAR T product, the disease being treated, and as well as the genetics and immune experience of these patients. But all of these patients, almost all of these patients, are experiencing some level of CRS response. When you look at all 4 grades, like a grade 1 is a minor CRS response up to grade 4, which is a very serious CRS response, we see that between 58 and 93% of these patients are experiencing some kind of cytotoxic response or CRS-related response. And a very high percentage, around 13 to 46%, are reaching grades 3 and 4. So a lot of these patients will end up in intensive care and being treated with steroids, tocilizumab, and other things to suppress that immune response. So what is this cytokine release syndrome exactly? Well, it can happen not only in immunotherapies, but also in infectious diseases. And we all know very much too well from COVID-19, these patients that are ending up on ventilators. This is because the infection has overstimulated the immune system. There's this cascade of signaling response that ultimately leads to vascular leakage, their lungs filling up with fluids, and they end up on ventilators. Well, monoclonal antibody therapies that stimulate an immune response, bispecifics, and CAR T can also lead to this cascade of response. And so these initial cells release these cytokines, which interact with other cell types that are part of the immune system, but also very importantly, there's interplay with the endothelial cells, which highlights the need for an in vivo system. In other words, there are in vitro assays out there that can be used to model some of this response, but they incompletely remodel it because not all the cell types are there that are involved with the response. And here's an example of that. So in the context of CAR T therapy responding to a CD19 B-cell lymphoma, initiates the cytokine release response. And other cells, like myeloid cells, can respond to these cytokines being released, release a number of cytokines of their own, which can also interact with the endothelial cells. And the endothelial cells can release IL-6, IL-1, GM-CSF, von Willebrand's factor, angiotensin II. That is this cascading response. You get this vascular leakage, and in the worst case scenario, you have immune cells infiltrating the brain and having an immune effector-associated neurotoxicity, et cetera. So in our mind, this is the argument for why a better assay includes an in vivo model that can measure all of these cell players in the cytotoxic response, but also to measure the consequences of this response. So even in the in vitro assays, you can measure the cytokine release, but what are the consequences of that? What does it mean to the tissues that are surrounding this? And we'll come back to that more a little bit later. So let's go through some examples that have been reported in the literature, so how people are using this technology and assaying that in these in vivo systems. Now, here's an example of taking a look at trying to target more than one tumor-associated antigen, looking at both CD19 and CD123. CD123 is the IL-3 alpha protein that's found on many cells. So what these researchers took a look at is that with just CD19 CAR T, you could clear out a lot of the tumor, but sometimes these patients would bounce back, or they would still have tumorigenic cells in their system that could be recognized through a genetic approach through fish or something like that. And what would happen is that these patients, before they received CAR T treatment, the tumorigenic cells were CD19 positive and CD123 double positive. But after being treated with the CD19 CAR T cells, they still had tumor cells that were CD19 negative, but CD123 positive. And so what they did is they took a look at tumor cells, and they were confirmed to be tumor cells, again, genetically by using fish, and look at those cells by CD19 and CD123. And what they did is they sorted out and isolated these CD19 negative, CD123 positive cells and injected them into an NSG mouse, followed them over time, and found that they acted like stem cell populations and recapitulated the CD19, CD123 double positive cell population. So these were kind of the leukemic stem cell population, explaining why there was a rebound following the CD19 CAR T. So what they wanted to do was develop an approach by dual targeting both of those markers to see if they could reach better tumor clearance. So what they're doing is they're comparing mice that are engrafted with NALM6 B-cell leukemia cell line or a patient-derived B-cell leukemia, then treating with CD19 CAR alone, CAR 123 alone, or a pooled combination of those two CAR T populations, or a CAR T that is expressing both CARs specific for both the 19 and the 123. And that's shown in green here. And what you can see, interestingly enough, those CAR T that were dual expressors did better than pooling the CAR Ts, both in the CDX model, but also in the patient-derived model, was much more efficacious. Here's an example looking at folks examining affinity tuning, so the strength of the binding of that extracellular domain of that CAR construct. And so what they did here is they took several different CDX models and just looked at the expression of HER2, because these are HER2-specific CAR Ts here. So some of these tumors are very high expressors of HER2, and some of these tumor models are low expressors of CAR2. And the reason for identifying low expressors is to use a cell population that will model the normal cells within the human patient. So what they're trying to do is the tumors that are high expressors would be the target of the tumors and killed, and then we were looking at off tumor but on target effects with the low expressors or the normal tissue expressors. They developed two populations of these HER2 CARs, the high affinity versions and the low affinity versions. And what they found is both the high and the low affinity versions bound to the high expressors. And they assayed this by mixing them in culture and measuring the response of these CAR Ts by their activation responses by releasing or upregulating CD107 or releasing interferon gamma. So they did it in two different assays and found the same thing. The high and low affinities were binding to high expressors, but only the low affinity, only the high affinity receptors bind the low expressors as shown here. So these are the two high ones, the solid bars and the pixelated, and they're binding to those low affinity ones. So then they took this to the next level to do the in vivo assays, which was a pretty neat trick. What they did is they took the SKOV, which was the high expressing ovarian cancer, and put it on the left flank of the mice, and the low expressing PC3 and put them on the right flank. And then they treated them with the high dose, high affinity, or high affinity low dose, or the low affinity CAR T at high dose or the low affinity at low dose, and looked at the tumor responses. So with the high dose, high affinity, it cleared both tumors. The high affinity, high dose mostly cleared the SKOV high expressor, and partly cleared the PC3. And so that was considered negative because what that would mean is that it's off tumor on target toxicity responses. So in other words, normal tissues also expressing HER2. So that would be a toxic event. Even with a high affinity at low dose, it cleared both again. And what was really nice here is that the low affinity CAR T at low dose cleared the high expressing tumor, while the low expressing tumor, which was an example of the normal tissues, persisted, uh, showing that the balance between affinity and the dosage of the CAR T could lead to more appropriate targeting of the tumor tissue. Here's an example of improving CAR NK. And this is CAR NK mediated B cell lymphoma model. So they're targeting CD19. But one of the problems with CAR NK is persistence. So in other words, if you take NK cells that are engineered to express these CARs and put them back into humans, the persistence of those CARs are low. Now, NK cells are supported by the expression of interleukin 15. One of the initial approaches to improving the longevity of CAR NK systemically is to treat mice with interleukin 15. The problem with that is interleukin 15 injections is highly toxic and leads to a lot of damage of tissues and decreased survival. So the approach here is to co-engineer these human NK cells to express IL-15. So it's a self-expression of the CAR and the IL-15 to improve the durability of these NK cells. But just in case there was some kind of toxicity mediated by these CAR NKs, they also included expression of an inducible caspase 9. That's what the IC9 is here. So what they did is they engrafted the mice with tumors and then injected the CAR NK cells. Then they isolated cells from the broad bone marrow, liver, and spleen, labeled those cells with human CD45 monoclonal antibodies and looked by flow cytometry. And what this would do is would label both the human NK cells as well as the human tumor cells. So use that as the human gate and then asked, well, what is the persistence of the tumor cells by looking at CD19 by flow cytometry? And in the IC9 CAR19 IL-15 NK cells, very nicely cleared out those Raji B cell CD19 positive tumors. So they're cleared right out. And the other CAR that was introduced that was without the addition of the IL-15 did not clear those tumors. And by looking at CD56 labeling, of human CD56 labeling, we could see good persistence in the presence of the IL-15 expression and the absence of those NK cells after 21 days in the mice, showing that there's a lack of persistence and the lack of clearance of the tumor. And then in those mice that had persistent development of those NK cells, if you treated those mice with a small molecule that leads to dimerization of these caspase 9 molecules, you get this dimerization and you induce this apoptotic pathway. You could actually clear these CARs out. So it's a very interesting set of technologies and able to introduce these cells. Now, one of these things to be noted in the same study is that with a single dose of the IC9, CD19, IL-15 CARs, there were some mice that persisted in terms of tumor development with extended time periods. So what they took a look at is a second dosing of the CAR NK cells. And what they found is something that was very interesting. One of the reasons for going to the CAR NK was this perceived notion that there would be less of this cytokine release response compared to CAR T using CAR NK cells. However, when they doubled up the dose, they found improved clearance of the tumor, but the sick mice in the survival assay also showed very high TNF-alpha, IL-1 beta, and IL-18 expression. So there is a cytokine release response if you give too many CAR-NK cells. In the third example we've got here, we're looking at HER2-specific CAR-M. And if you'll remember your ADCP biology, or antibody-dependent cellular phagocytosis, that's also mediated through CD16. So the CD3 zeta portion of that molecule can also activate the myeloid cell compartment. So the CAR construct is actually quite similar. You have an extracellular domain that binds your target. You've got the swivel domains and the membrane domain, and then you've got this intracellular domain that can activate these cells, and you can use CD3 zeta to do that. Now, what's also interesting about this study, with myeloid cells, what do you use for the vector delivery component? And many myeloid cells are resistant to infection by various adeno-associated viruses and various subtypes. And they found a very specific serotype of adenovirus called the AD5F35 that was able to deliver their cargo. And by transducing these cells with this adenovirus, you converted these cells to an M1 pro-inflammatory phenotype. And they took these cells and mixed them with their SKOV HER2 expressing cells in vitro and showed very specific lysis using that molecule. So that's the in vitro assay, and then they moved to an in vivo assay. And in this particular model, they used the NSG SGM3 model. This is the model that we have at JAX that is expressing human stem cell factor, GM-CSF, and IL-3. Now, this is important because these are human-specific growth factors that support human myeloid cell engraftment in these animals. So they engrafted these mice with the SKOV tumor cells and then injected IP into these recipients. And they treated these mice with either untransduced myeloid cells or CAR-transduced myeloid cells. And they could measure suppression of tumor growth. It wasn't successful in all of the mice, but there was a significant portion of those mice that had very nice suppression of tumor growth and extended survival. Now, they took this a step further, and they wanted to ask the question, since these cells have an M1 phenotype, if these cells go into the tumor microenvironment, can they change the immunophenotype of other existing infiltrated immune cells to accelerate the ability of T cells to respond against the tumor, et cetera? They did an interesting experiment where they took these NSG-SGM3 mice and they took female human hematopoietic stem cells and grafted those into the mice to establish a human immune system into these mice. Then it takes about 3 months for that engraftment to establish. They took the same tumor cells and put them into the flank of the mice. So this is a solid tumor model. They waited about 19 days so that they could allow the immune cells that are circulating in the mice to infiltrate into the tumor. Then they took their engineered myeloid cells and they used male engineered human myeloid cells, created those CARs, and injected them intratumorally. Now, the difference of male versus female would allow them, once these tumors had grown for about 5 days, to isolate these tumors and do single-cell RNA-seq to tell the difference between the hematopoietically derived and the CAR-derived myeloid cells, and also look at those immune phenotypes, whether they're M1, M2, et cetera. And just to make a long story short, what they found was that the introduction of these M1 CAR-M cells could convert the resident myeloid cells present in the tumor microenvironment to an M1 phenotype. And they also went on to show that it would allow these myeloid cells to cross-present tumor antigens and activate the T cells within that microenvironment. So very sophisticated, very interesting experiment. So in summary, Dual targeting of cancer-associated antigens may prove very useful to address antigen loss, mediated tumor relapse, or the problem with heterogeneous tumors. Co-expression of cytokines in so-called armored CARs can enhance the persistence and tissue penetration of these introduced CAR NK populations. Suicide genes can be used as a safety switch to manage toxicities due to cytokine release or due to on-target off-tumor activities. And all these technologies are very, very interesting. CAR-T, CAR-NK, and CAR-M show great promise, but they all do have safety issues that must be carefully addressed to balance efficacy with some of these cytotoxic responses. So JAKs and a number of researchers here have recognized the importance of having a better assay to model this difference between tumor efficacy and the cytokine release response. Now, there are model systems out there for looking at these different things independently, but as I said earlier, what are the consequences of cytokine release that are measured in an in vitro assay? What is the duration of the cytokine release? The magnitude of the cytokine release, and what does that mean to cells and tissues within the organism that lead to toxicity? So in our mind, it would be better to do this in vivo when everything is happening at once, and you can look at the balance between the two and assay everything in the same system. Now, to get away from concerns of human T cells being introduced to the mouse that could lead to GVHD, we've created a mouse that has mouse MHC class 1 and class 2 knocked out. So we can put tumors in, introduce PBMCs, then introduce the CAR and look for those responses. So the goals of this CAR-T mediated assay is to develop something that's fast, sensitive, reliable, and reproducible. The idea is to de-risk these therapies for preclinical development. Far too many of these promising therapies are being terminated in clinical trials due to the toxicity events that develop from them. And so what we would like is for this novel assay to give holistic toxicity and efficacy data in this multi-organ and multi-tissue responses that really try to mimic the human pathophysiology. So we're trying to develop a sensitive assay that includes PBMC humanization and do this in a very short assay. And I'll show you that this can be done within a 2-week assay. You need to include both the cellular targets for the efficacy and the appropriate immune cell types that can measure the cytokine release response, and then look at the balance between efficacy and cytokine release. And very importantly, there are donor-to-donor differences response, as I showed earlier, that are found in the clinic, and we need to model those differences in response. And we can see in this model donor differences in response, okay? And we need to be able to look at drug combinations and things like that. So the mouse model that we're using for this, as I mentioned a moment ago, is knocked out for mouse class 1 and class 2. If you compare the engraftment, we're just putting in PBMCs, mice that are irradiated, either the class 1, class 2 deficient mice shown in green or a regular NSG mouse. You irradiate them, put in human PBMCs, and then interrogate those mice for what's engrafting. You can look at the total human CD45 and then ask what proportion of those human CD45 cells are CD3 positive T cells, CD19 B cells, CD14 myeloid cells, CD56 NK cells, or CD33 positive myeloid cells. And all are being engrafted. There's primarily an expansion of T cells, but these other subpopulations are found in these mice. If you use body weight as a measure of the development of graft versus host disease, you could see that in the NSG mice, they need to be sacrificed because they start losing weight very quickly, whereas the longevity of these mice is very nice. The double knockout mice, stay alive and healthy for a longer period of time. And this is a survival curve as shown here. So this gives you a model to measure these in the absence of severe graft versus host disease. It doesn't eliminate graft versus host disease, but it very significantly delays it. And this is an example of looking at 4 different PBMC donors and the engraftment kinetics in terms of their percent survival. So you can overwhelm the system. So in other words, if you double up the dose in the double knockout, you can start to see mice that will develop graft versus host disease. And some do this worse than others and some less than others. And these 2 are the regular NSG mice that routinely develop graft versus host disease in the system. So what's important about this is we are looking at the influence of the donor cells in this reaction. And we have a panel of PBMC donors that we've pre-characterized and know what cell dosages can be introduced into the system. So this is just simply looking at CD22 CAR T and its delivery into the system and its response and ability to clear out Raji, Raji B-cell lymphomas. And it's also important to include a mock CAR T group because these are human T cells that are expressing their native TCRs, and you don't want to look at tumor clearance based on an allogeneic response against the tumor, and that's not the case. It's only the CD22 expressing CAR Ts that are clearing out the tumor by less luciferase expression. So you're looking at this is just a graphic representation of the previous slide showing the clearance of the tumors. And these mice are maintaining body weight quite nicely. It's only those with high tumor burden that are beginning to lose body weight in this model. You can also interrogate these by injecting human PBMCs, coming in with your CD19 CAR T, and using the resident B cells from the PBMC injection and looking at clearance of those B cells as your efficacy portion of the model. And you can look in the whole blood and you can look at the spleen, and you could look at this across 2 different donors in this particular experiment. The efficacy response was similar in terms of clearing out those human B cells by the CD19 CAR T in both of these donors. But interestingly enough, if you look at the cytokine response, there is a cytokine response of interferon gamma, TNF alpha, IL-10, and IL-6. But what's interesting here is between these 2 different donors, the 9534 and the 9531, very big difference in the amount of IL-10 secretion within these 2 different donors. And you can tie that back to things like body weight change as an indicator of a global or in vivo or tissue cytokine release mediated cytotoxicity. These mice that received this particular donor, the 9534 with the high IL-10 expressor, showed loss of body weight, whereas the 9531 did not. We have an internal built-in control into this sample where we've got treatment of those mice with anti-CD3 OKT3, which we know from previous experience and published reports that leads to a cytokine release, which is transient. And that cytokine release that's transient leads to a transient body weight loss, and the mice bounce back from that. So there's donor-to-donor differences in response that can be identified. In this particular experiment, we're co-injecting human PBMCs with the idea of what is the influence of amplifying that response, because it's the additional T cells that are involved in the environment that are involved in the cascading response. And also, what are the effects of increasing the dose in this context of the CAR T, going from 1 to 3 to 5 million cells? In this particular donor, we did not see body weight loss as it's clearing out these tumors in these mice. But we did see good tumor response, and we saw a dose response from the CAR T in terms of tumor clearance. The body weight decline was only in those setups where there was just PBMCs, or excuse me, no PBMCs at all, or only PBMCs in the absence of CAR T. But in the presence of CAR T, as you increase the dose, you increased the interferon gamma response or release. So there was a CRS response here. So what we can see here is that the inclusions of PBMCs is important for amplifying the cytokine release response, as you'll see in a minute. So one thing that's important to note that if we look at the blue, the tumor growth is the same. The blue is the inclusions of PBMCs alone is not clearing out the tumor. It's only when you have the presence of the CAR T in either the green or the orange. And the orange is the combination of the PBMC and the CAR T, and you have the similar clearance of the tumor, and this is statistically significant at days 10 and at day 12. But what I want to point out here is what I said a moment ago about the inclusion of the PBMCs to amplify the response. So again, there's no cytokine release when you add PBMCs in the absence of the CAR T as shown in blue, either 24 or 48 hours after introduction. But if you compare the green, which is CAR T alone, which introduces release of cytokines, interferon gamma, TNF-alpha, IL-10, and IL-6, if you include the additional PBMCs, you amplify or increase the cytokine release response as you would in a normal human immune system where there's all these other ancillary immune cells around. So we've looked at this in the context of monoclonal antibodies, CAR T, bispecific antibodies. These are very rapid assays. They're very tight assays. They're very reproducible. The number of mice per arm can be kept down to 4 or 5 mice per arm. There's a lot of ancillary readouts that we can use to look at the consequences of this cytokine release, body temperature, immunophenotyping to look at the activation state of these cells. We can look at cytopenias that are a measure of activation-induced cell death, clinical chemistries, looking at liver toxicities, histology for infiltration of these cells. And we have evidence that once you have this type of activation, you can look at histology and immunochemistry to identify those cells and tissues, et cetera. So in summary of chapter 3, this NSG MHC double knockout mouse can significantly delay the onset and severity of xenogenetic graft versus host disease, enabling more extended studies of this model. The platform can identify donor-specific differences in the magnitude of cytokine release as observed clinically. Increasing doses of CAR T can amplify cytokine release in the presence of a tumor target, was exactly what you would expect, and that's observed clinically as well. The inclusion of the human peripheral blood mononuclear cells provides an additional human immune cells that are capable of increasing the sensitivity of the cytokine response, and that's very important in this assay. The in vivo analysis is necessary to accurately evaluate the toxic consequences of tumor lysis and cytokine release. So JAX, in conclusion, JAX has developed what we think is a very, very nice in vivo, systemically holistic, rapid, and flexible platform to accurately, and we think more accurately, assess both the efficacy and safety within a single assay. The JAX platform can assist researchers and drug developers kind of really take a look at the complexities of CAR therapy. That you really need a platform to look at these things like binding affinity, the construction of the transmembrane domain and spacer domain, the co-stimulatory and activation domains, co-expression of cytokines, chemokines, checkpoint inhibitors, combination therapies, all of those different things. And there's the ability now to look at all of these different cell populations, CAR-M, CAR-NK, CAR NKTs, all these different things, and look at the tissue penetrance, longevity, et cetera. So there's a lot that this assay can do. And so many people think of JAX as, a mouse house and producing these different models, but we perform CRO activities. We have an expert team that can run these types of studies. They have expertise and experience in running these models. These mouse models sound simple to run, but there are a lot of complexities in terms of the timing of how to run these assays. And since they've developed these models, they really know how to run them best and most efficiently. So I need to acknowledge the folks that have developed these assays. They're part of Jim Keck's group and his team. Of course, Lenny Schultz, who has created a number of really important models that we use in our CRO activities, and then our collaborators, Mike Brem and Dale Grainer, down at UMass Med. Thank you very much, everybody.